My name is Raheem, and this is the research that I've been doing in the Toronto lab for the past few years. The research in the lab is mainly organized around the study of naturally occurring N-N bonds, which are rare in nature. Here, we see CreE taking nitrite and incorporating it into our 3,2,4 amber to create creamycin, which gives us an N-N bond. Creamycin is an antibiotic produced by Streptomyces creameus, and antibiotics are made by bacteria to kill off other bacteria. The nitrite that is being used to create our creamycin cannot be added to the cell culture. It has to be created by the cell, or there would be no creamycin made. This is the actual purpose of the research, which is to study the mechanism of CreE. CreE takes L-aspartic acid and oxidizes it 3 times, giving us a 6-electron oxidation by a single enzyme, which hasn't been seen before. CreE uses oxygen and NADPH, which is an electron carrier, to oxidize L-aspartic acid into nitrosuccinate. After the oxidation of L-aspartic acid into nitrosuccinate, we have CreD come in and cleave off the nitro group, giving us the nitrite that is being used in the previous slide by CreM to make creamycin. To study the mechanism of CreE, we must first produce its protein, and that's done by taking competent cells, which are bacterial cells that can undergo a genetic transformation, and transforming them with plasmid DNA. Plasmid DNA is a circular piece of transferable DNA. The plasmid, which contain our gene CreE, is inserted into the competent cells by heat shocking them. The heat shock allows the plasmid to enter the bacterial cell. After the heat shock, there is a recovery period where we give the bacteria time to produce antibiotic-resistant proteins encoded on our plasmid. Once those proteins are produced, antibiotics can be added to the cell culture to prevent any other bacteria from growing in the culture. After there is a significant growth, we add IPTG, which induces protein production. When enough protein is produced, the cell culture is then centrifuged and lysed, and the lysed solution should contain our protein CreE along with all the other proteins produced by the cell. To get our protein CreE from the lysed solution, we ran it through a nickel support chromatographic column. Proteins interacting with this column has an engineered sequence that gives them 6 histidines at the end of their amino acid chain. These 6 histidines bind to the column, and they are the histidine tag. The column has competitive interactions with our histidine tag and imidazole. When adding the lysed solution, which contains our protein, and all the other proteins in the mixture would flow through the column, and our protein, which has the hist tag, will bind to the column, and it will not be released until rinsing the column with imidazole. Washing the column with the imidazole would be considered our elute, which is where the proteins are supposed to be. But we did not get any significant bands in our gel showing the protein exists within our solution. There was, however, a band for our whole cell fraction, where we took the cell culture after induction, centrifuged it, and chemically lysed it with loading buffer, centrifuged the solution again and diluted it, then directly placing it into our gel. This gave us our whole cell fraction, which expressed that our protein exists but was insoluble. For the protein to be soluble, a significant band must be seen in the lysate. There were alterations in our protocol to give us soluble protein. Initially, we did transformation with BL21 cells, which were lysed by sonication, but that did not give us any soluble protein. We then moved on to LEMO21 cells with a lower temperature of incubation and with the addition of L-rhamnose, which slows down transcription to give the proteins time to fold properly, where proper folding of the protein allows them to be soluble. None of this gave us soluble protein, so we thought that the proteins CreE and CreD may be codependent and moved on to trying to insert them into the same plasmid. To insert CreE and CreD into the same plasmid, we must first amplify the gene by PCR, which stands for polymerase chain reaction. PCR works by designing primers that can anneal to a gene of interest, allowing the polymerase to come in and make complementary sequences of the DNA from the 5 to 3 end. This is a series of steps where our reaction mixture is heated and cooled, denaturing the DNA and allowing the primers and polymerase to come in. After about 3 steps, we would get our amplified gene segment. When designing primers, we must encode for restriction enzyme cut sites, which would show as overhangs on our amplified gene. These overhangs are considered sticky ends that could be bound into our plasmid. The plasmid would have the same restriction enzyme cut sites, allowing for complementary sequences from the primer with the restriction enzyme to bind into the plasmid with the same restriction enzyme cut site. To insert CreE and CreD into the same plasmid, we must use a PET duet vector, which has 2 multiple cloning sites. Initially, we had CreE in PET 26b, which only had 1 multiple cloning site. As expressed before, CreE must have its primers designed to encode for the restriction enzyme cut sites, which are shown here by NCOI and PSTI. Most of the work has been done with CreE, but once the amplification is successful, we will then move on to the amplification of CreD and its insertion. Throughout many attempts to amplify the gene segment CreE, we did not get any amplification. We changed various things, such as the polymerase use, where we use fusion, which is a high fidelity polymerase, and GOTAC, which is a low fidelity polymerase. We had primers designed, as mentioned in the previous slide, and T7 primers, which are primers specifically designed to sequence PET vectors, where all PET vectors have the same T7 promoters and terminators. Even with the addition of HF, which is high fidelity buffer, and GC, which is supposed to be for a high GC content, and the addition of magnesium chloride did not give us any of our amplified gene segment. There was another gene that we had in the lab that was also in a PET vector. Using common PCR ingredients and the T7 primers, we got amplification of that gene, suggesting that there was probably something else wrong with our gene segment. After looking further into our gene, we found that CreE had a high GC content, as seen here where all the G's and C's are highlighted blue. The high GC content would prevent proper denaturing of the DNA in steps of PCR. The GC nucleotides increase the melting temperature of the DNA because they have 3 hydrogen bonds between them versus AT, which only has 2. With the addition of DMSO, the melting temperature of the DNA is lowered, allowing for proper denaturing within the PCR reaction. This gave us amplified gene, which can be seen at the 2000 kD mark. With the successful amplification of CreE using GOTAC and DMSO, We then tried using fusion, a high-fidelity polymerase, with the addition of DMSO. Using fusion with DMSO did not give us our amplified gene segment, so we thought to order different sets of polymerases to see if they could amplify our gene. The polymerases ordered were Q5 and platinum Taq. Both polymerases used gave us our amplified gene segment. Platinum Taq successfully amplified the gene, but not as well as Q5, where we have Q5 give us a heavier band. With the successful amplification of the gene CreE using Q5, we then tried to use Q5 in mutagenesis reaction to delete the 4 histidines as mentioned before, with primers designed for them. Initially, when trying to amplify the gene CreE, we did not get any amplification. When we first sequenced the gene, we only sequenced one side of it, and that side matched perfectly with the native CreE DNA sequence, which suggested we had the right primer design. But after sequencing the other side of the gene, we saw that there had errors. On the top part of this slide, we have the bottom half of the native CreE protein sequence. On the bottom part of this slide, around the RLTEVTA region, there were 4 extra histidines found that should not be there. It is possible that these 4 extra histidines might be encouraging improper folding, which may have caused some issues with culturing the cells and trying to extract our soluble proteins in previous steps. With the deletion of the 4 extra histidines, we would have to amplify the whole plasmid, which was about 5,000 base pairs, plus our gene CreE, which is about 2,001 base pairs. With the use of DMSO, amplification was successful with a clearly visible band. There were a different set of primers used for this reaction that needed to be phosphorylated because the amplification of the whole plasmid would produce blunt ends that needed to be ligated together. The protein production in the initial steps of the research was hindered by having insoluble protein, which may have been caused by the 4 extra histidines and or the need to express both CreE and CreD into the same plasmid. All the failed attempts of amplification seem to have been mainly caused by the high GC content of CreE. With the addition of DMSO and the use of the T7 primers, there may have been successful amplification, but the amplification also seems to be dependent on the polymerase used, where we could not get any amplification with fusion, but amplification with GoTac and Q5 were successful. After amplifying CreE and deleting its 4 extra histidines, the next steps would be to amplify CreD and to insert it into a PET duet vector along with CreE, where this new plasmid can be transformed into competent cells to see if the production of CreE is a codependent one. The amplification of CreE with its 4 histidines deleted will also be used to transform competent cells to see if the removal of those 4 histidines would encourage proper folding. Thank you for your time, and I hope you enjoyed this presentation. I would also like to thank Dr. Caranto for giving me the opportunity to study in the lab.